So during mitosis or meiosis, which is cell division, the cell goes through something called the cell cycle. So this part right here with checkpoints G1, S, and G2, that's interphase. And the one I want to focus on is the S phase, where DNA synthesis replicates the genetic material. And this is when the single chromatid becomes two sister chromatids. Okay, so during DNA replication, the enzyme helicase will split the DNA strand in half, and then single-strand binding proteins will hold the DNA apart. And then primase, as you can see on this strand, will synthesize the red RNA primers to initiate DNA replication. And DNA polymerase III is found both on the lagging strand and the leading strand. And DNA elongates in the 5' to 3' direction. So here at the replication fork, the leading strand, it could continuously add to the 3' side nucleotides. But over here on the lagging strand, it has to add going in this direction, 5' to 3', then stop, go back a little, add some more 5' to 3', and the DNA polymerase I, which is over here, is going to remove these RNA primers and replace it with the matching DNA nucleotide. And then at the end, DNA ligase will join these fragments of DNA, which are called Okazaki fragments, to connect it into one segment. And at the end of replication, there's something called telomerase, which will add short DNA sequences that don't really affect the code itself. Next, I want to do an overview of transcription and translation. The central dogma biology focuses on is DNA creates the mRNA, then mRNA creates the protein or polypeptide chain. And in bacterial cells, this all happens in the cytosol or cytoplasm. And as you can see in a eukaryotic cell, transcription and RNA processing is within the nucleus and translation happens outside. So for transcription, the first step is initiation. And this is when the RNA polymerase attaches to the promoter region. And it begins to unzip the two strands, and it often contains a sequence called the Tata box, which is thymine and adenine. And then next, during elongation, the RNA transcript molecule is formed from the enzyme here. And termination is when RNA polymerase reaches the end sequence of nucleotides and terminates the sequence. And then here's a visual of the RNA polymerase, which is putting RNA nucleotides against the template strand of DNA. So the mRNA codons are going to have the exact same sequence as the complementary strand, with the exception of the uracil instead of a thymine. So you could see some of the nucleotides match up right here. And then there's also transfer RNA. So the tRNA is this thing right here. It has 3 anticodons, and it matches up with the mRNA codons and an amino acid will attach right here to form a growing chain of polypeptides. So here you could see the strand of mRNA and the 3 codons getting attached to the tRNA, and then a unit or a polypeptide attaching on.